So good evening. Basically, see, some of the recent developments are related to like certain aspects of science and technology. Again, mm, see, there is a lot of development that happens every time in the various aspects of science and technology, whether it may be defense, biotechnology, space, and all. Sometimes, identifying the right areas for the exam is also very, very important. Now, I just wanted to start with the say mRNA vaccine. Okay, I want to start with mRNA vaccine. So, what exactly is mRNA vaccine and why this has been in the news? So, again, see, from the beginning, as we say, there is a need to have some basic information regarding each and every subject, isn't it? See, what exactly is this mRNA? Hmm? And what is the central dogma of molecular biology? So once we have the clarity of the central dogma of the molecular biology, we'll be able to understand clearly what such things easily. And tomorrow you can identify the options, the right options in the question in the preliminary exam, or even any issue related to the mains can be asked. So can be answered very perfectly. Now, so why we are talking about mRNA vaccine? What is the reason? See, in fact, recently the Pune-based Genova Biopharmaceuticals, the Pune-based Genova Biopharmaceuticals has been given a restricted use permission for its mRNA vaccine. So India's first indigenously developed mRNA vaccine. So what is that? India's indigenously developed mRNA vaccine. For what? Huh? Vaccine for which particular disease? So for COVID-19. So what we are discussing is mRNA vaccine for COVID-19. Now, basically, what is a vaccine? What is a vaccine basically? So vaccine is a preventive medicine, isn't it? Vaccine is a preventive medicine. So when do you take vaccine? Before you get the disease or after you get the disease? Both. So basically, we take the vaccine before we may, get the disease, and that's why many people, had been given this vaccination, COVID-19 vaccination, and the universal immunization program, which is being carried under Mission Indradhanush, also provides vaccines to kids and pregnant women. Yes, there are some chances where, say, if you are bitten by a rabid dog, imagine if you are bitten by a rabid dog. Now, if you are bitten by a rabid dog, that is a dog with rabies, now immediately you should be able to get a vaccine. So against the rabies. So in certain conditions, and not only that, if you are injured, if you are injured, then you will be asked to take what? TT, tetanus toxoid. It's a toxoid type of vaccine. So you will not get that particular tetanus if you take today after injury. So vaccines are really very great in terms of preventive, because they are preventive for many diseases, and various vaccines were given as a part of COVID-19. And say India's indigenous Covaxin is there. So what type of vaccine it is? So it is an inactivated vaccine. What do you mean by when I say it's an inactivated vaccine? So when I say it's inactivated vaccine, it very clearly indicates that the virus, the SARS-CoV-2 virus, is completely killed. It's killed and introduced into the body. So what you are taking in Covaxin is a dead virus. A dead SARS coronavirus is what you are taking. So, in inactivated virus. What is the advantage of introducing an inactivated virus into your body? What exactly is the advantage? Why should we introduce such a dead pathogen? So because if you introduce a dead pathogen, for example, like what we are talking about, SARS-CoV-2, so this is dead now and you have introduced, so it will not cause any disease. Then in such a scenario, since it doesn't cause any disease, so then the body, the immune system will get triggered and starts releasing antibodies against the antigen of the virus. What is the antigen? What is an antigen? See, antigen is a foreign substance generally present on the surface of the pathogen which triggers your immune system. See, if a pathogen enters your body, how do you think that the body will identify that particular pathogen? See, some X bacterium, X virus enters your body. Now, don't you think that this particular pathogen should be identified by your body? What happens if a pathogen, X pathogen enters your body and then it's not identified by your immune system? What happens? What's going to happen? It will create havoc. It will destroy your body systems. So definitely immune system should identify. How can it identify? It can identify by special surface properties the pathogen exhibits. Special surface properties. Say imagine. So take any virus, take any bacterium. Imagine this is a bacterium. Its surface, the cell wall is made up of so many different types of chemicals. Do you think that every chemical present on the surface of a pathogen will also be there in you? Do you think that the surface molecules present in any bacterium, virus or any such pathogen will also be there in you? No. See, hence some of these substances, that's why I said foreign, which is not a part of your body. So these chemicals which are a part of the surface of the pathogen, which are not found in your body, will be triggering immune response, because your immune system identifies this part of the pathogen as a foreign substance and hence it starts attacking it. So that is, this entire pathogen can be identified as a foreign entity by just observing this particular chemical which none of your cells have. None of your body cells have this chemical. So definitely your immune system can identify what is called self and non-self. That is a major role of immunity. Major characteristic of immune system is what? The ability to distinguish between self and non-self. So as soon as a pathogen enters, remember, this part of the virus, this part of the virus or this part of the pathogen, so which is not a part of your cells, not a part of your body, immediately will be identified, and yes, this is foreign, and the one having it is also a foreign. So immediately immunity will be triggered. How it will be triggered depends again on an elaborate process. Now one simple way to say is, on identifying the pathogen through this foreign substance, your immune system starts releasing missiles, and these immunological missiles are given the name antibodies. So not only that your immune system releases antibodies, but also keeps memory of that particular antigen and the pathogen. So that if the next time the same pathogen enters, that memory helps the immune system to identify it as the one that has come earlier. So this time what will be the response? Second time if the same pathogen enters your body. So what is the response? It will be very quick and more number of antibodies are released. It's very quick and more number of antibodies are released. That is a system on which your immune system actually, that is what the principle on which immune system acts. And that is the principle called immunological memory, based on which we are developing today vaccines. So why? Because imagine, so in the case of say inactivated SARS, that's corona vaccine, like Covaxin of Bharat Biotech. So imagine this part of the spike protein, this particular spike protein is acting as what? Is acting as an antigen. So you have introduced a dead virus, then the dead virus cannot cause the disease. The dead virus cannot cause the disease, but your immune system will identify this as foreign and starts what? Producing missiles and developing memory. And the second dose, what happens? The second dose that you have taken, or any booster dose that you will take, so what happens? The second dose will enable the body to produce more and more antibodies and increase further the immunological memory. So that next time if the same pathogen enters, your immune system will easily destroy it because it has been seen by the body already twice. So this is what the principle is. Now, rather than see which part of the virus or which part of the pathogen should be shown to the body in order to trigger immune response, which part of the pathogen actually is triggering immune response? Antigen is triggering immune response. So then why you are introducing the entire pathogen? What if your inactivation is not done properly and the virus is still live? What if your inactivation is not done and the virus is still live? Instead of preventing the disease, it will cause what? Disease. So hence my point here is, the modern vaccines started, introducing only the antigen separately, like what we call recombinant DNA vaccines. But let me mention the pathogen, the SARS-CoV-2 here has a special spike protein or S protein which acts as an antigen. So, what is antigen chemically? Chemically, the foreign substance, this or any such foreign substance on the pathogen, foreign substance on the pathogen, which triggers immune response. What is it chemically? Was it chemically? So chemically it may be anything. But what is the ultimate principle here? Ultimate principle is it should be foreign. It should not be a part of your body. And the second rule is what? The second point is about it can be anything. Chemically it can be anything. It's not only a protein. It can be a protein, it can be a lipid, it can be a glycoprotein, what not. It can be anything. Glycolipid also. Okay, and remember, there is another rule in immunology, and let me, before going to that rule, if you remember whatever just now I said, that an antigen can be chemically anything. So imagine a situation where this particular pathogen has so many foreign substances on it. Do you think that all of them are acting as antigens and triggering immune response in your body? See, there are so many. This is not a part of your body. This chemical on the surface of the pathogen. This is not a part of your body. This is not a part of your body. But they're all foreign. That's what I'm saying. They're all foreign to you. But do you think that all these foreign particles on the surface of the pathogen really trigger your immune response, or do you think that all of them act as antigens? Now comes a point. To act as an antigen, the foreign substance should have a molecular weight fairly above 10,000 daltons. So fairly above 10,000 daltons. So daltons, unit that used to measure this atomic weight. So what is that? How much? Fairly above 10,000. So these are the principles. Tomorrow if you get a question in preliminary exam, so this is how they try to give you the options here. So like earlier, way back, there was a question in civils prelims with relation to antigen is a substance, A, B, C, D, triggers foreign substance that triggers immune response. That is the option. So here, if they want to give a question on vaccine, if they want to give a question on, say for example, Covaxin or the one which we are talking about, mRNA vaccine, they will use the word antigen in one or the other way. In such a case, how do you answer the question if you don't have some idea about what exactly is antigen? And remember, The knowledge on antigen and antibody alone will help us in understanding the immunity and the so much knowledge of the vaccines. So hence the base is very, very important. Okay, coming back, what I am trying to say is, in the case of uh, like SARS-CoV-2, SARS coronavirus, now the antibodies are released against an antigen on its surface, which we are calling as S protein, spike protein. So the point is, what if I introduce the protein directly into your body rather than the pathogen? Rather than the entire pathogen, what if I release only this particular protein against which your body releases antibodies? Got my point? What if I do that? Then your body starts producing antibodies. That's recombinant DNA vaccine. So where I can make only a protein, there is a process. But here, remember, for the production of any protein in your body, even your body produces a lot of proteins. For the production of any protein in your body, the thing is, there is a process where the protein information for the synthesis of the protein. Tell me any one single protein that is there in your body. Give me any idea about a single protein that you have in your body. Any protein that you have right now in your body. Somebody was saying, yes. Keratin is one protein that is present in your nails and hair. Any other protein? Melanin is not a protein. It's a pigment, but chemical, it's not a protein. Yes. That's it. Only keratin. There are all proteins. See, your body is protein. So whatever if you remove the skin, what is there around your bones, muscle? It's made up of myosin, actin. What is the saliva contains? A protein by name tyelin. Your eyes, the eyes contain an enzyme called what? Lysozyme, which is antimicrobial, antibacterial specific. That's a protein. So protein. Now your keratin is synthesized because that information is there in your DNA. That information in DNA is copied in the form of what is called, is copied in the form of something, an intermediary called messenger RNA. What is that? Messenger RNA. That RNA takes the message for the synthesis of your keratin out of the nucleus and gives it to the ribosome. The ribosome reads the information on the RNA, which I am calling mRNA, and accordingly it makes a protein. The ribosomes, they read the information on the RNA and make something called a protein, and that protein comes out of your cells of the root hair as what? As keratin. This is how it comes out. So this protein keratin is synthesized by whom? Ribosomes. And who carried the information from the DNA to the ribosomes? mRNA. So who has actually the information, for the synthesis of keratin? DNA. That part of DNA which has information for the synthesis of a particular protein is gene. This is what gene is. Gene is. So what we have to do is, if coronavirus has a spike protein which is acting as an antigen, what the Genoa biopharmaceuticals or any other such mRNA vaccine, what they can do is they will make such mRNA, they genetically engineer mRNA which has information for the synthesis of what? Which has information for the synthesis of that spike protein. So what if I introduce genetically engineered mRNA into your body, which has information for the synthesis of what? The coronavirus S protein. What happens? I genetically engineer that mRNA in such a way that, again I repeat, mRNA will have information for the synthesis of what? A protein. So I just taken an example. So I will genetically engineer mRNA that has information for the synthesis of which protein? Corona SARS virus 2 spike protein, which acts as an antigen and which triggers immune response when the coronavirus enters your body. So what I'll do is, so that genetically engineered mRNA I'll introduce into your body. Now that as soon as it enters your body, it is designed in such a way that your muscle cells especially pick up that and synthesize what? Synthesize what? Imagine your muscle cell. The muscle cell has picked up what? mRNA, and they synthesize. Who is there in the cells to synthesize protein based on information in mRNA? Ribosomes. So ribosomes synthesize, and this is the spike protein. Imagine spike protein. Now this spike protein released into your body, into your bloodstream. So start stimulating what? Start stimulating your immune cells to produce antibodies and memory. Thereby 2 times I'll give you in 2 doses. Then finally, if really SARS virus enters your body, the textbook rule, which I am trying to say, if really SARS virus enters your body exhibiting its spike protein, what about your body? It already had seen the spike protein twice and has given so much antibodies and also developed immunological memory. This time, will you get the disease or will you destroy the virus? That's it. As simple as this. This is a very simplified version of the DNA mRNA thing. There is a lot to basically, this is a very simple form of a simple way of explaining. It's not just DNA to mRNA to protein, as such it happens. So that this is a simplified way of understanding. So the basic subject is different again. So the information in DNA copied into RNA is called transcription. And the information of the mRNA copied, the information of the mRNA studied and made a protein by ribosomes is called translation. So this is what exactly DNA to RNA, RNA to protein is the basis of life on the planet Earth. Is the basis of life on the planet Earth. And what is this DNA to RNA? Just now I said, the method of information exchange that happens, the process in which the information in DNA is studied for the synthesis of RNA's transcription, and this is translation. So this is how basically we have to understand. Once we have this idea, automatically people, once we have this idea, after studying that mRNA vaccine has come up, you will be getting clarity. You will be really getting good clarity on that. Okay, understood that? Now, so hence again coming back to the real information that, so for the first time, like India has come up with Gemcovac, right? So Gemcovac, India's first mRNA vaccine for COVID-19. Gemcovac 19. So India's first indigenously developed mRNA vaccine for COVID-19. India's first indigenously developed vaccine for COVID-19, mRNA vaccine for COVID-19 is Gemcovac 19. Developed by Pune-based Genova Biopharmaceuticals. Genova-based biopharmaceuticals. Okay. Now, further, actually, see, it's not that we are the first, in fact, to develop mRNA vaccine. But the point is, the mRNA vaccine developed in US and other parts of the developed countries, so it has one major problem, that it has to be stored at sub-zero temperatures. So the mRNA vaccine should be stored at what? Sub-zero temperatures. But the speciality of our vaccine here is that the company has achieved thermostability for it, so that it can be active even if it's stored in standard refrigerator temperatures. Standard refrigerator temperatures. Standard refrigerator temperatures. So hence, please try to understand. The vaccine, such mRNA vaccine developed by the US and other countries, so they don't have this ability to be active at the standard refrigerator temperatures in the medical shops, whatever you call. So at the but they have a condition where they can be active only when stored at sub-zero temperatures. So indicating that this is another important thing, because we'll have greater adaptability. Tomorrow, India has an ability to further export it to various countries where where there is no such provision of storing at sub-zero temperature. So hence, this is one unique achievement that India has done. And whatever this GEMCOVAC has been given as given restricted emergency use approval for 18 and above years of age. Restricted emergency use approval has been given to GEMCOVAC. Okay, so this is what. So hence, please try to understand. So don't just restrict your information, your knowledge to the news, which is in fact very danger. Okay, so apart from that, big thing happened and everybody was mesmerized to see the images provided by James Webb telescope. I think you followed that in the newspapers. So నేను మళ్ళీ చెప్తాను. So what we were discussing, let me discuss, right? Now the point is, I was talking about what? Yes. So I was talking about what is called the James Webb telescope. So if you look at the James Webb telescope, now the point is, what exactly is the speciality of the James Webb telescope? What exactly is the speciality of the James Webb telescope? First of all, why man is really interested in studying the origin of universe? Why man is really interested to study the other planets? Why we are trying to collect information beyond our solar system, beyond our galaxy? What's the reason? See, there should be. And are you not identifying something like a space race across the world today? Space race. Say people they are trying to get onto the Mars. Okay, and Nambi Narayan was recently talking that India had an ability, would have been first. And even there are great chances for India to go and land on Mars first. Yes, it's not that don't underestimate Indian space strengths. The only thing is we have big crunch of human resources, so funding, so which are very very important. And India of course requires a separate policy, separate what is called process by which it can speed up this also. So which one? The Martian exploration, manned Mars mission. So we had already in the form of a Mangalyaan. What is that? Unmanned Mangalyaan mission we had. Orbiter. So where we have launched an orbiter onto the Mars. Now, having said that, so let me also mention this Hubble telescope. And tomorrow, see, there is a chance for a question in the mains exam. Even it may be in the general essay, or even in the GS3 also, there may be a question with relation to like this. So there is an increasing interest. There is an increasing interest across the globe to explore universe and our solar system than ever before. So based on that, you can be asked, like, discuss the discuss the reasons for such, exploratory activities, and their significance. So this type of question is possible, because it looks at a broader picture rather than at the micro level single program objective. So it will have, a greater thing. So it's not like, with relation to just exploration of universe. It may be with relation to exploration of Mars, for example. So because renewed interest and especially the Perseverance, rover, and all these regarding Mars, pictures about Mars, information about the mineralogy of Mars, all that, and trying to identify any indicators of what life on Mars, and now extending our what is called ability to gather information from the distant parts of the universe. So these are the things which need to be covered in such a question. So there is a chance for such a question in the exam, mains exam, and even very important for the even generalists also. Now, coming to the James Webb telescope, James Webb telescope. Now, first of all, it was named after James Webb, who was the second, NASA administrator and worked as an Undersecretary of State in US government and contributing much to the, space science. So James Webb is second administrator of NASA and contributed much to the development of, space science in USA. So hence, now the James Webb telescope is an extension to what? Is an extension to, universe exploration that was already carried out by, that was being carried out by whom? Hubble Space Telescope, carried out by already Hubble Space Telescope. Hubble Space Telescope. See, this Hubble Space Telescope was launched in 1990, whereas the James Webb Telescope was launched in 2021. So now I'll try to, compare both also. First of all, where do you, put a telescope if you want to study the sky? Where do you arrange a telescope if you want to study the sky? Where exactly you want to arrange this telescope if you want to study the sky? Is that you arrange on the road? No, you'll try to take a high level. Because of nowadays light pollution, a lot of glare, created is really preventing these budding enthusiast astronomers or in general professional astronomers to study the space, because the glare of the light pollution coming from the light pollution is preventing them from visualizing a very clear sky. So hence we try to go at the higher levels. What if we put the telescope into the space? This is what it's. So we try to put a telescope into the space, then it becomes what? Space telescope, so that we'll get a clear, picture of what the entire universe. So that's the reason. And really, do you think that India has such a telescope? Space telescope, or India is developing such a space telescope? So India in fact has launched already a space telescope called Astronomical Observatory Satellite. So India's space telescope is Astronomical Observatory Satellite. Astronomical Observatory Satellite. Astronomical Observatory Satellite. It was launched 2015 September 28 on board PSLV C-30. Now when it was launched, people said India's Hubble Space Telescope. Now remember, don't compare this because it's a very small scale telescope. Which one? Our AstroSat. So hence we should not compare. Even ISRO also representatives clearly said that please don't compare AstroSat with Hubble at that time. So but it is also collecting a lot of information, hmm, on from distant galaxies, regions, and all. Okay, in the universe. Now coming back. So first of all, where this particular James Webb telescope has been launched. Actually, the Hubble Space Telescope is moving around the Earth. So remember, it is moving around the Earth, not away from the Earth. So whereas the James Webb Telescope is moving around the Sun. So making Sun as a center. Now, in the Earth, Sun, Moon system. So you know that Sun is here. Then imagine this is Earth, and imagine this is what? Moon. In the Earth, Moon, Sun system, you should have certain areas where you can park your satellite spacecrafts so that you can have a distant view of the Sun. Like for example, Aditya L1, which we are going to launch. We are going in launch into the L1 point. And where this, was launched? Which one? Where this was launched? The James Webb Telescope. The James Webb Telescope, for example, yes, has been launched into the L2 point. L2 point. So this is the place where James Webb Telescope has been launched. See, these points were provided, to park the satellites in such a way that they will not be affected much by the Earth and Sun forces. And Joseph Louis Lagrange was the first person who had identified a mathematician who had identified such spots, and hence they are named after him. Okay, so they are in fact named after him.